Hello, this is Miss Dakenford, and we're going to be looking at eukaryotic gene regulation, which is regulating protein synthesis. And if you think about what happens throughout the day, throughout the year, we've talked about how animals and plants respond to the coming of winter, hibernation, and all of the differences that happen within the cell. Well, those don't happen by themselves. They have to be dictated by proteins, which of course come from the information in genes. So think about all the different signals that cause this to happen. They could be the length of daylight, the temperature, chemical signals like insulin and testosterone and plant hormones that all tell the cell you must suddenly make this or stop making this. So how does that happen? Now, if you wanted to, you can hit pause and go ahead and copy down what I have here, and then we'll come back and examine what all of these mean. Now, this diagram does a really good job outlining where we're going to go. We're going to talk about the chromatin modification, the genes that are available. So that's going to lead to transcriptional controls and then mRNA processing. And then how do we then manipulate how they go outside, exiting the nucleus? And then what happens to the protein after it's translated, whether or not it's degraded or sticks around a long time? How does it get processed and then outside? At every single one of these steps, the cell can regulate whether or not that protein gets out and does its job. Well, let's go ahead and dive right in. Let's take a look at chromatin. Now, chromatin is, in essence, DNA. It's what we call the long strands of DNA as it's packaged. Now, chromatin normally is packaged with histones, but we can manipulate how tightly these histone proteins come together or how spread out they are. So let's go back to this idea of packaging the chromosomes. If you have this really tightly packaged genetic information, and as this DNA is packaged, well, if we want to get at a gene that's like right in here, well, it's all hidden. But what if we were to loosen that a little bit? And so let's use the same picture. But what if that gene that we want gets unraveled and then the rest of it, of course, stays? So if that's the gene that we want, well, now we have access to it. And so there's ways that we can do that. And it has to do with a lot of chemistry of our molecules. So we'll come back to the cat in just a second. So there's two ways, and it has to do with the different chemical groups that we put on our histone proteins. Now, the histone proteins are the spools that the DNA strands are coiled around. And notice they have these little tails that stick out from them. Those tails can be manipulated by what groups that get added to it. So you can have acetylation, and acetylation is when we put acetyl groups on it. And acetyl groups push away from each other and it loosens the chromatin. So instead of being all bunched up, now you have an open access. Now, we came up with a couple of years ago this mnemonic that acetylation activates that gene, mainly because we now have access. If I scroll down a little bit, you can see the second part of this. This over here, well, you're starting to kind of see like a chromosome, like a thicker structure. But if we open this up, now we have the gene that we need. And these are acetylated histone proteins, and it just causes it to push away from each other. This can now be transcribed. Well, the opposite can also happen. We can methylate. We can put methyl groups on, and that causes the chromatin to clump together. And as it clumps together, it shuts within itself and it silences that gene, mainly because we don't really have access to it. Things can't get to it. It truly is a physical phenomenon. Everything just kind of closes up. So we came up with methylation mum's the word. So you get the idea that it's silent. Now, a really good example of that would be the chromosomes in mammals. Well, in female mammals, you have two X chromosomes. Well, what if one of those chromosomes told the cat to make black fur and the other chromosome told it to make orange fur? Well, what will happen is because there are two X chromosome, one gets very, very strongly methylated and it condenses so tightly that you just kind of see it as this black smudge under a karyotype. And if that's the case, then that one is silent. Now, what's really wild is that for female mammals, and this includes humans, but cats, we see it a little bit cooler. What X chromosome gets silenced in a cell is different from cell to cell. So especially at an embryonic level, so if we had an embryo, a little clump of cells that's turning into a cat, and this cell, let's say this cell is now, they have the X chromosome that has the orange gene turned on, well, you're going to get a clump of cells that will have an orange spot. But let's say over on the other, on another cell, you might have a gene that's saying, no, make black fur. Well, then that might correspond to this region. And that's why we get that calico pattern. So we can close and open the chromatin. Now, that's one way. But what about telling the cell to actually transcribe? And this is really wild. This is one of the most fascinating areas of research lately in the gene regulation. Uh, the transcription initiation in eukaryotic cells is just even more complicated than the operons. But I do think it's a little bit more visually intuitive because it's all about attracting that RNA polymerase. And so in the transcription initiation, we have these upstream elements. So here's our gene that we want to transcribe. And upstream, we have our enhancer regions. These are DNA sequences that will attract activators. And these activators, I know it just like keeps going and going, right? So these activators are going to be specific transcription factors that, and other proteins that will bind to those enhancers. We're going to look at what happens. Now, this is a nice way to practice. We've got our DNA and then mRNA. So think right here should be transcription. DNA is transcribed into mRNA. And then mRNA is going to be processed here. We're going to splice out those introns, and then we're going to get our mature mRNA strand. So how does this work? Well, let's take another picture. In our typical upstream elements, we have our activators, which are proteins that are going to bind to the enhancer regions. Well, so what? I kind of hinted that already. Well, the so what comes when we realize that this can actually bend over and you have a specific binding protein that helps with that. And these activators are going to form this connection between the enhancers and the transcription factors that allow RNA polymerase to then know this is where I go. It's a very strong signal to tell RNA polymerase to transcribe here. Now, what really puts our picture together, though, so here we have our picture that basically now says transcription is now going to happen. Notice the arrow. We've got our transcription factors, our activators, our enhancer regions, and RNA polymerase. Now it connects. It's just going to zip along and transcribe that DNA. Well, that's all well and good, but why do we care? Well, we care because this now gives us an amazing insight into how cells can dictate what proteins they make. So let's take a look at two different cells. And one is going to be an eye cell, the cornea, and then one is going to be the liver cell. And we're going to look at two different proteins. Two different genes that are going to make two different proteins, the albumin and the crystalline gene. Now, the liver is going to make albumin, and the I is going to make crystalline. But this is the important thing. Both DNA sequences are in liver and eye. Remember, unless you're a red blood cell, every cell has a nucleus with all of the chromosomes that we need. And so the liver has genes for making an eye, but they're silent. Well, how come they're silent? What happens? Well, you're going to have different activators in the different cells. So here's our liver cell on the left and eye cell on the right. And notice the big deal here is the activators are now different. You've got a pink one, orange one over here, and a gray one. And yes, the same gray one over here, but notice a red and a yellow. And so these are going to bind to the correct enhancer sequences, cause that to bend over. Here are transcription factors, boom, 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 RNA polymerase, transcribe. But notice the crystalline activators are absent. We don't have these here, so the crystalline stays silent. Over here on the eye cell, we've got all of our lovely crystalline activators, which boom, 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 combine to the transcription factors, combine to the enhancer region, bends over. Here's our RNA polymerase, transcribe the crystalline gene. So you want to think about, remember I've been talking about how do we know this? Well, we mess with it. We combine. What if we take away this activator? What if we mutate these enhancer regions? What if we mutate the crystalline gene? What if we suddenly lose the red? Or what if suddenly this enhancer region mutates so that it accepts the red activator? So all of these are really intriguing questions, and we want to address them. So that gives you a really nice example of how eukaryotic cells start regulating transcription.